I went there at Delhi for a friend's wedding. Okay. So I visited ICGEB, my lab, Dr. Nayer's, and then all the PMB labs actually.
Dr. Nair is
now they are individual. Yes, he is still there.
Okay.
He is still there.
And Dr. Sapur is also there? No.
Professor Sapur is there, but as an honorary or something.
Emeritus, yeah, he has joined as emeritus scientist. Okay.
Yeah, he's there. Snehalata ma'am was also there, so it was a good thing. But to be frank, he will not like a bit because I feel now it's not really, you know, like well coordinated. Before one group leader, so there was some control. Now everyone is a different coordinator.
Everyone wants to tell their own story.
Yeah, that's it. And also the labs and space are not clean. It's so dirty. I was so surprised because I took all my friends from here and they wanted to visit ICGEB.
Oh, but so, that's shocking to hear.
Yeah, seriously. It was like normally.
I've seen the young clad is not that good in being disciplined in all these things. So maybe the PIs are a bit normal now.
Yeah, maybe. Maybe today was better for us. For people like us.
For Dr. Snehalata, it is not so easy.
Yeah, no.
Amit, I know Amit used to be so particular about everything. I was like, oh my God. I used to get complex when Amit used to work. I was like,
okay. Now we are in live, so.
I'm sorry. We just engrossed in our
so you can't mention any name now.
Yeah, okay.
But you can chat. You can chat. Yeah, no problem for that.
We can just do one practice. You can share your screen once and then we can stop your screen share for a moment. But we can just try once that it's happening fine.
So is that okay? You can see my share. Right?
Yeah.
I made the slides in a very general way because I don't have that much of data from my side to put in. So mostly it's kind of a review paper or something like that.
Okay. No work on glutathione? Nothing?
Oh, I have. At the end, I have some examples.
Oh, that's fine. We just do. It was just to see that this screen share is working fine. So Soma will be introducing you today. Pardon me for today. Generally I do it, but I'm not that well today. So I'll be here for a few minutes, and then I'll be keep on coming and seeing the chat and the Q&A box for discussing questions and all those things. But no verbal contribution from me today. I can't do that today.
No problem. I hope I will do justice for that because I'm a bit nervous, to be frank. Like,
don't be nervous. You would have seen the other recordings.
Yes, I have seen. But the thing is, after seeing them, I'm much more concerned now because they are very prominent in something. So I still consider myself
answer round. Whatever you feel comfortable, you can pick up those questions. It's not necessary for you to answer all the questions. Whatever question you feel you are able to answer, you are comfortable with, you answer pick few of them and you answer them. You will be seeing the Q&A box on your own, right? After the talk, just go through it. We'll be dismissing irrelevant questions, but sometimes they just keep on coming while you're seeing the chat. So don't worry about that. You can answer based on your time, and if you have time, you can answer more questions that way.
Okay.
I know you have a meeting just after that.
I want to say something to Tahmina. Just remember that you are the boss, and you know everything, and nobody knows, and nobody else knows anything. Just remember that.
So that's the main point, I think.
Yeah, I hope so. Let's see. And unfortunately, I have a meeting exactly at 10. Like professor scheduled, he actually forgot that I have an appointment and he is scheduled with other partners, so I couldn't cancel that. But I will try to finish on time so that we can finish it.
Yeah, sure. And even I have to run to make a cake for my daughter's birthday.
Okay.
Oh, that's why you are unavailable. Today is 17th.
Yes.
Oh, happy birthday to Lily.
Thank you.
It's your daughter?
Yeah.
Yeah, please convey happy birthday from my side as well.
Thank you. She's 3 today, and I just put her down to sleep. She was like clinging to me and just put her down.
That's good. That's good. So,
When do
okay,
we
we are.
start?
It's three
12.
4 minutes remaining.
Okay.
So you have prepared your presentation for like an hour or 45 minutes or how?
40 to 45 minutes. Yeah, around it will take 40 to 45 minutes.
Yeah, we have ample time. You see, you can speak slowly. You want to explain whatever you will do. We have ample time. You do according to what is comfortable to you.
I think Neha's internet connection. Yeah, okay, it came back.
Where does Shubho went?
No.
Yes.
I am here. I'm always here. Okay, I almost did everything. Today we got registration. Today we got registration almost not exactly 58 countries all over the world. And I asked Soma if possible then name few of it, because sometime it's important to others or listener also. And as I know that talk is very brief and exactly on the time, so we have little bit 2 minutes to describe the name of these countries. We got 3,200 something registrations.
Okay.
So I mailed about 99% because some email address are error and G Suite's not accept their email addressing. But we put your and our YouTube channel link to the website. So those are interested, they definitely come here to see you. So I'm going to broadcast this session to allow the attendees to come in the Zoom.
Okay.
Okay. Time now.
Okay, it's 12, so we can start. Welcome everyone to our Bioingene live webinar. This is the 7th webinar in the series, 8th webinar in the series, and Soma will introduce our speakers. And Soma also doing the question answer round for today, and interview round also. So I hand it over to Soma to welcome our speaker. And I also mentioned that don't panic about your certificate, because it's very annoying when you start asking the link before the talk. We are giving the certificate for the webinar, not for your registration, not for your show off in that boxes. So please listen the listen the webinar, learn from it. During the question answer round, I will give the link as well the password. And I absolutely want to assure you that you get the link and password at the exact and appropriate time. So no, don't ask many times for the link, because we miss the relevant questions during that chat box. So please cooperate with us. If you give the feedback, your name will be in our website, and you get the certificates. But be patient. So I hand it over to Shoma. Sorry, Shoma.
Okay, hello.
Hello. Welcome to today's webinar organized by Bioingene. Bioingene is a non-profit organization created to promote plant science research worldwide. The webinar series was designed to build a platform from where plant scientists can present their research to the world. We hope future scientists can gain perspective and inspiration by listening to the esteemed plant researchers talk about their scientific accomplishments and their thoughts on the future of plant sciences. We are grateful that many renowned scientists have accepted our invitation to share their research insights with us. We are also thankful to the audience for their interest in bioengine webinars. We have received a huge response of over 3,000 registrations for today's webinar. You can attend the webinar through Zoom webinars and view live on our YouTube channel. Video recordings of today's event will be available on our YouTube channel. Please subscribe so that you don't miss out on any of the webinars. It is our request to all the viewers to please hold all questions for the question and answer round at the end of the presentation. You can type in your question related to today's talk in the Q&A box. You can apply for a certificate of attendance via the feedback link and password, which will be provided after the presentation. I repeat, the feedback link and password will be provided after the presentation. So please be patient. Today, we have viewers from all over the world, from 58 countries. People have registered from 58 different countries, from Afghanistan, Algeria, Australia, Bangladesh, Belgium, Benin, Botswana, Brazil, Cameroon, Canada, Chile, China, Colombia, Czech Republic, Ecuador, Egypt, Ethiopia, Germany, Ghana, Greece, Guyana, Hungary, India, Indonesia, Iran, Ireland, Italy, Japan, Kenya, Korea, South Korea, Lebanon, Malaysia, Mali, Mexico, Morocco, Namibia, Nepal, Nigeria, Pakistan, Peru, Philippines, Poland, Russia, Saudi Arabia, Senegal, Somalia, South Africa, Sri Lanka, Sudan, Tanzania, Trinidad and Tobago, Tunisia, Turkey, United Arab Emirates, the United Kingdom, and the United States. We welcome you all. We are very glad that you have registered from all over the world. The topic for today's webinar is the role of reactive oxygen species and antioxidant machinery in crop plants by Dr. Tahmina Islam, Postdoc Fellow, Plant Breeding Institute, University of Kiel, Germany. Okay, so let's see what we have here. We have Dr. Tahmina here today. Dr. Tahmina Islam worked as an assistant professor at the Department of Botany, University of Dhaka, Bangladesh. Currently, she is doing her postdoctoral research in the Plant Breeding Institute of University of Kiel, Germany. Her main research focus is plant stress biology and genome editing. She has completed her bachelor's and master's from the Department of Botany, University of Dhaka. Her PhD dissertation focused on abiotic stress tolerance in rice, which she completed from the International Center for Genetic Engineering and Biotechnology, New Delhi, India. With her work, she aims to develop stress-resilient crops in context of the South Asian subcontinent, specifically Bangladesh. I now request Dr. Islam to start her talk. Ma'am, could you please share your screen now?
Yeah, sure. I hope my screen is visible. Shoma, is it visible?
Yes, it is visible.
Yeah, okay. Thank you very much. I would like to start my presentation, but before starting that, I would like to thank you all from the Bioingene team to give me the opportunity to showcase some of my research work, and also it is a very great platform for the plant scientists to basically for showcasing their work, and also we are learning so many things. So far, I have attended some of the seminars, webinars in the YouTube, and I find it very, very helpful. So very good teamwork from you guys. So with this, I will start. So good afternoon, everyone over in India and other countries. Good day. So today I am going to talk about a research project that I did during my PhD, but I will talk about in a very simple way so that everyone can understand what is the role of reactive oxygen species. So the title of my research today is Role of Reactive Oxygen Species and Antioxidant Machinery in Crop Plants. First of all, I will definitely talk about the reactive oxygen species itself, and later on I will move on to the mechanism, how it works behind, and also I will discuss about some examples in crop plants where plants can definitely tolerate this kind of situation over the time. So to start with, I will start with a brief introduction about the reactive oxygen species, which are partially reduced and excited forms of atmospheric oxygen. Those are really named as superoxide anion, peroxide, hydrogen peroxide, then hydroxyl radical, etc. So according to the literature and also from the different research, reactive oxygen species actually function in cells as signaling molecules, but are also known as the unavoidable toxic byproducts of aerobic metabolism. From the evolutionary point of view, ROS, that means reactive oxygen species, most likely appeared on Earth together with the first atmospheric oxygen molecules about 2.4 to 3.8 billion years ago, and have been a constant companion of aerobic life ever since. So highly reducing condition and also highly levels of soluble iron in the atmosphere or the primordial oceans, it is possible that most of the atmospheric oxygen initially produced was almost immediately converted into ROS over the time. Here you can see they're converted into ROS. So the evolution of aerobic life on Earth therefore occurred in the presence of reactive oxygen species. And this is the fact that we should be kept in our mind when we consider the roles of reactive oxygen species that currently play in different biological systems. So how the major reactive oxygen species generation happen or occur in cells? In particular, here you can see the superoxide anion, hydrogen peroxide, and also hydroxyl radical can be produced in nearly every cellular compartment. The singlet state of oxygen is generated mainly by the energy transfer from triplet state of the primary electron donor or photosystem. One electron reduction of the oxygen and photosystem 1 produces moderately reactive superoxide, that is this superoxide anion here, and which can be dismutated to hydrogen peroxide by SOD, that is superoxide dismutase, in the presence of further the transition metal, that is iron and copper. Further reaction takes place and to the hydroxyl radicals. So this means that this is a long chain of formation of ROS generation, and every steps could be very detrimental to the plant. For example, superoxide anion, hydroxyl peroxide, hydroxyl radical, everything could be very detrimental to the plant health in this. And the lifetime of these radicals are varied from microsecond to nanosecond, and which is completely different. And I will talk about each of them in coming slides. So the type of the ROS, first I will talk about the superoxide radical. This is a very brief introduction because this is necessary to know how detrimental could be this ROS actually. So this is a superoxide radical is the first reactive oxygen species to be formed. This is mainly formed in the thylakoid localized photosystem 1 during the non-cyclic electron transport chain, as well as the other cellular components. Moderately reactive with a short lifespan of 2 to 4 microseconds. It doesn't cause that much of extensive damage by itself, but rather than it undergoes transformation into more reactive and toxic hydroxyl radical and also singlet oxygen, and that can cause the membrane lipid peroxidation. It is reported that in the plant system, it could be scavenged by superoxide dismutase. Next, the singlet oxygen. It is an atypical superoxide, atypical ROS actually, which is generated by not by electron transfer to oxygen, but rather by the reaction of chlorophyll triplet state in the antenna system of oxygen. Singlet oxygen can cause severe damages to both of the photosystem, that means photosystem 1 and photosystem 2, and that's how it puts the entire photosynthetic machinery into the jeopardy. Even though singlet oxygen has a very short half-life of about 3 microseconds, but it could even very detrimental and could diffuse some 100 nanometers and cause damages to a wide range of targets. These targets actually include the molecules like proteins, pigments, nucleic acids, and lipids. Major reactive oxygen is responsible for light-induced loss of photosystem 2 activity elicited the cellular death. But plants have managed to efficiently scavenge the singlet oxygen with the help of beta-carotene, tocopherol, plastoquinone, and can also react with the DI protein of the photosystem 2. Basically, non-enzymatic antioxidants are responsible for the scavenging of singlet oxygen in the plant system. Upregulating genes, which are responsible for the providing protection against the photostatic stress, that can be also upregulated by the singlet oxygen. So singlet oxygen also can work as a positive way, as a signaling molecule. So that is also possible in case of singlet oxygen. Next is a hydrogen peroxide, which is moderately reactive oxygen. It can occur both in non-enzymatically by being dismutated to hydrogen peroxide under low pH condition, and also by mostly a reaction catalyzed by superoxide dismutase. Electron transport chain in the chloroplast, mitochondria, endoplasmic reticulum, cell membrane, beta oxidation of fatty acid, and photorespiration could be the possible sources of this hydrogen peroxide generation. It has significantly half-life to 1 milliseconds compared to the other ROS members and can cross membranes via aquaporins and cover considerable length within the cells. 50% loss in the activity of different enzymes, and also responsible for the programmed cell death at high cellular concentration. It is reported that the hydrogen peroxide, which is moderately reactive, can be scavenged by several antioxidants, and those are enzymatic antioxidants, for example, here, catalase and peroxidases, and also non-enzymatic antioxidant flavonoids take part into the scavenge process of hydrogen peroxide. Lastly, hydroxyl radical. This is the mostly active and most toxic known about among the family members of this reactive oxygen species. It is generated at neutral pH by the Fenton reaction between hydrogen peroxide and superoxide anion, catalyzed by the transition metal like iron. So lipid peroxidation, protein damage, and membrane destructions are the most symptoms of this hydroxyl radical destruction in the plant cells. So far, no existing enzymatic system to scavenge this toxic radical. Excess accumulation of a hydroxyl radical can cause the cellular death as well. Mostly it is scavenged by flavonoids and proteins. What could be the major sources of ROS into the plant cells? Mostly the photosynthetic machinery is the most vulnerable, I would say, most vulnerable machinery actually to the plant system to be attracted or to be the target of this reactive oxygen species generation. So in the presence of light, mostly chloroplast and plasma. Chloroplast mainly become the source of this or ROS production into the plant cell. On the other hand, in presence of dark, that means in the dark condition, mitochondria become the major point of ROS production into the cell. So I must say, include about the peroxisomes as well. In case of light condition, peroxisome also could be the one major source of ROS generation. And so what happened into the mitochondria during the ROS generation? So plant mitochondria is basically known as the energy factories of cell and is one of the major sites of ROS production, such as hydrogen peroxide as well as the other sources. It differs significantly from the animal counterparts with specific electron transport chain components and also function in processes such as photorespiration. Cellular environment of plant mitochondria is also distinctive because of the presence of photosynthesis. And because of the photosynthesis, it creates an oxygen and carbohydrate-rich environment into the plant cell that is completely different from the animal system. So here you can see the complex 1 and complex 3 of mitochondrial electron transport chain are very well-known sites of superoxide anion production. In aqueous solution, the superoxide anion is moderately reactive, but this ion can further reduced to hydrogen peroxide by the distribution by the dismutation of SOD. It has been estimated that about 1 to 5% of mitochondrial oxygen consumption leads to the hydrogen peroxide production. So this hydrogen peroxide can react again for reduced with iron and reduced to produce highly toxic hydroxyl radical. And this hydroxyl radical is very detrimental and which can really penetrate the membrane and leave the mitochondria. Mitochondrial level is also, I must wait. Yes. The mitochondrial ROS level are crucial for the biological outcome. Actually, in 2011, Hamanaka and Shandel, they proposed a model which basically shows the mitochondrial ROS level are crucial for biological outcomes. Low level of, here you can see, low level of mitochondrial ROS production are required for cellular process like cell proliferation and differentiation. And induction here in the ROS production will lead to the adaptive programs, including transcriptional regulation of antioxidant genes. Here you can see that there is an induction of ROS here. So later on, even higher levels of ROS will signal the initiation of senescence and apoptosis. But the non-signaling here, the non-signaling and it is irreversible damage to cellular components is only observed under the highest level of cellular ROS. So in that case, there is no chance of for the plants to overcome this kind of damage into the plant cell. So which is the highest detrimental point where plants are unable to fight back. So next, the ROS generation into the chloroplast. In higher plants and algae, photosynthesis takes place in chloroplast, which contains a highly organized thylakoid membrane system that harbors photosynthetic apparatus. Oxygen generated in the chloroplast during photosynthesis can accept electrons and also can produce, here you can see, also produce the superoxide anion. Therefore, the presence of ROS producing centers as the triplet chlorophyll and also the electron transport chain in photosystem 1 and photosystem 2 makes the chloroplast as a major source of the ROS production. This superoxide anion can further dismutate to hydrogen peroxide, as I mentioned before, which is moderately active and can damage the cell. Thirdly, we will discuss about the ROS generation into the peroxisomes. So what is peroxisome? It is basically an organelle which are very small, usually spherical macrobodies bounded by a single lipid bilayer membrane. There are subcellular organelles and probably the major sites of intracellular ROS production. In the peroxisome, two sites of superoxide anion production are established here. First one is in the organelle matrix, where xanthine oxidase catalyzes the reaction or the oxidation from xanthine to uracil acid. The second site of peroxisome into the peroxisome membrane, where a superoxide anion produced, and there, which is actually NADPH dependent. And in that process, MDHR and also cytochrome B participates in the formation of superoxide anion. The main metabolic process responsible for the generation of superoxide, generation of hydrogen peroxide actually in different types of peroxisomes are the beta oxidation, then the photorespiration, purine metabolism, sulfide detoxification, sarcosine metabolism, polyamine catabolism, flavin and oxidases, and of course superoxide dismutase. There are some other organelles which are responsible for ROS production into the cells, so although these are not really very familiar and also not the major sources, but they have some potentials, or there are some few attentions nowadays are giving to those organelles which can produce ROS into the plant cells in other organelles. For example, apoplast, then also the plasma membrane. So here you can see in the plasma membrane that is possible, it is possible to produce the reactive oxygen species here, and also outside of the plasma membrane, H2O2, that means hydrogen peroxide, can produce extracellular spaces in the apoplast. So it is also reported that the function of this hydrogen peroxide in apoplast is pH dependent. Basically, alkaline pH promotes the ROS production into the apoplast. As I mentioned before, that other organelles, for example, endoplasmic reticulums, lysosomes can be also a source of ROS production. So here you can see there are some production of ROS here, and here this iron is responsible for that. So you will see in my coming slides also, this iron, the labile ions are into the cell, actually creates a lot of problems for the plants, because this could be a source or could be I would say a factor which really initiate the formation of reactive oxygen system into the plant cell. So far what we are known about the thing about what is ROS and how it generates and the sources of the ROS, but there are some important facts about ROS that I can say that regardless how and where they are generated, an increase of in intracellular oxygen result into two very important effects. Either it could damage to the cell or can activate specific signaling pathways. So both of which influence the numerous cellular processes leading to proper cell function or to the cell death. And there are also so many reports and studies regarding that ROS were originally considered to be exclusively detrimental to the cells. But it is now recognized that the redox regulation involving for reactive oxygen species is the key to the modulation of critical cellular functions as well. Some of the pleiotropic effects are really harmful in case of reactive oxygen species, but they have many roles as signaling molecules in addition to their scavenging protective function in biological system. For example, I can say here, antioxidant gene modulation, redox status, calcium mobilization, then ion transport, host defense genes, then also host cell death, apoptosis, necrosis, cellular cross-linking, phytoalexin biosynthesis is also a part of the ROS signaling pathway. So basically it works in a both way. So we can really conclude in that way that ROS has a dual role in vivo depending on a different level of reactivity. So it really depends which molecules are working behind and their level of reactivity, sites of production, because obviously mitochondria and chloroplast are the major sources, and these are the important organelles for the plant cell as well. So sites also matters, and also the potential to cross the biological membranes. So if, for example, if the ROS or reactive oxygen species are not really, crossing the biological processes or membranes from one organelles to others, or not the cell membrane, then maybe it is not that much detrimental. But you can sequester it there, you can scavenge it there. It can possible, but if it has the potentiality to cross the biological membranes, then it could be really harmful for the plant cells, and that is very detrimental at the end. So as far as we discuss about ROS, its generation and other topic itself, it is also very important to know some other terms that is play a very important role in case of stress biology, which is oxidative stress and ROS network concept. So oxidative stress is basically the accumulation of reactive oxygen species at that level where it is beyond the capability of the plant cell to scavenge it. So plant cell cannot really, at the oxidative stress condition, plant really cannot scavenge the reactive oxygen system for this, from the cell. So at that stage, antioxidant and other scavenging systems really doesn't work, and it creates some stress for the plants. And on the other hand, if we talk about the ROS network, it actually is a compile of different genes, and I would say different proteins as well, and they work together to minimize the formation of ROS, or the minimize, not only the formation, also after the generation, it tries to detoxify the cell itself with this red network concept. So Foya and Shizuoka in 2010, they proposed a very simple scheme about the interaction between ROS network and oxidative stress. They described here that enhanced oxidation, here you can see enhanced oxidation, can lead to either programmed cell death or either enhanced sustainability. But it depends, as I also discussed before, it depends on the intensity of the oxidative signal and also the nature of the signaling pathway that is activated. That means if the intensity of ROS is really high, it will lead to the programmed cell death, or low reactive oxygen into the cell will lead to a defense mechanism signaling. So that will, in a way, it will help the plant cell to activate its downstream gene cascades to overcome this ROS situation. So I would say it is very well nomenclatured by Ron Mittler, who is a very famous scientist into the stress biology. He actually nomenclatured reactive oxygen species as a double-edged source of life. So it is, in one hand, it's very helpful, and in the other hand, it's very, very toxic for the plant cell. So with this, one question that came to our mind, that how toxic this reactive oxygen species could be. So because there should be a threshold, how much toxicity you can find there. So in 2017, Mittler, in one of his paper, it was a review paper, if I am not mistaken. So he discussed about a bacteria that is known as the Deinococcus radiodurans. And there he actually discussed about that this bacteria has the capability to survive in a very high toxic condition. That means in toxic condition means I want to mention about that high level of reactive oxygen species. But this property is not really unique to only this Deinococcus radiodurans, because there are some other bacteria also. They have found that they can also survive in the similar conditions. So now the second question comes, like how this organism can really survive on this such kind of highly toxic or highly ROS-induced environment. So there are several studies found after that, and they observed that certainly there are some factors which contributes for this kind of resistance on the ROS against the ROS environment onto the bacterial system. So if I make them in a very brief way, so first is the biochemical and cellular adaptation of that cell, then reducing endogenous oxygen, reducing endogenous reactive oxygen species, then actively ROS scavenging, and having a very efficient system to remove, replace, or repair damaged DNA and nucleotides and proteins from the system. And finally, sequestration of the cellular components, that is maybe the iron, as I mentioned before. So now you can ask me why really this bacterial system, and how can we really extrapolate this example into the plant system, because these are completely two different systems, I can understand. But it is possible. It is possible, and Mittler, in that review paper, he actually proposed a model, I would say, that in that model, he actually showed plant can also overcome this situation or overcome this toxic environment by activating some functions or adapting some functions. For example, here you can see this, the ROS can be produced by signaling path from the signaling pathways and also metabolically or externally. But this ROS can be controlled or maintained by scavenging with different antioxidant mechanism. For example, superoxide dismutase, catalases, peroxidases, glutathione peroxidases, etc. And also, as I mentioned, in the bacterial system as well, the sequestration is very popular or very common phenomena, I would say. And so these are harmful ions and can be sequestered into the vacuoles, so that it will not really harm or it will not really initiate any production of ROS. And also, if some damage can happen or damage can already occur into the system, that could be repaired by the DNA and protein repair system, and also protein and organ removal by autophagy, as I can say as an example, and also new biosynthesis. But there will be always some damage that cannot really, in the high toxic condition, you cannot really control all the cells to be damaged, but you can minimize the situation very well. So, but in most of the cases, you can see ROS leads to the signaling. So when it leads to the signaling, basically it initiates the downstream gene cascades to fight against the ROS. So which is very essential for the plant. So I would say, or it is better to say that because of the plant and animal cells have high ROS scavenging capacity, and the example that I presented here provide an evidence that reactive oxygen species, even at very high levels, cannot really kill cells. It is becoming more and more plausible that ROS-induced cell death is mediated through a programmed genetic pathway. So it can initiate it, but it is the programmed genetic pathway that leads to the cell death. So, and also it is not a result of the cell simply succumbing to an overwhelming ROS injury. Perhaps, I would say, like perhaps the emerging concept of redox biology that I will discuss in coming slides can provide more clarification to this question. So redox biology, what is redox biology and what can actually happen into this total system will answer our questions that we raised in this slide. So redox biology, if you ask me, or if I can try to actually define it, so it's a broad term used to describe the regulation of gene expression, metabolic pathways, and signal transduction mechanism by redox reactions mediated through the oxidation and reduction of cysteine residues of proteins that alter their structure and function. So here you can see that the peroxisome, mitochondria, chlorophyll, and cell-bound peroxidases result in the accumulation of H2O2, that means hydrogen peroxide or superoxide anion, into the cell, and that mediates the oxidation of cysteine residues into the protein, affecting their structure and function, and that triggers the cellular signaling pathways, here the signaling pathways. However, the presence of this labile iron, as I mentioned before, that it could be very detrimental or initiates a ROS, can change the situation in a way so that it leads to the production of hydroxyl radical. That will ultimately leads to the DNA damage, protein oxidation, lipid peroxidation, chemical cross-linking, etc. Ultimately produce the oxidative damage to the cell. So maintaining the cellular pool of labile iron, I would say here, as low as possible, therefore is very crucial for redox biology. And for the regulation of metabolism and other cellular function of ROS, this balance or this regulation is very, very important. So we will discuss this about in our coming slides also, but now I will give some examples as we discussing about the positive side of the reactive oxygen species. So I will give you some examples where ROS plays a very specific roles in a positive way in plant growth and development. So there are emerging evidence that ROS homeostasis control the plant vegetative apex form development. In this example is in Arabidopsis thaliana, where superoxide anion are basically accumulate into the meristematic region of the root tip and is required for cell division, whereas this hydrogen peroxide are basically important for the elongation zone and which confers the cell differentiation. These two different ROS microenvironments coincide with the meristematic and elongation zone, and their distribution are very important for the localization of the transition zone. Because of the ROS species gradient, cells entering into the transition zone can still proliferate. So once the ratio of superoxide anion to hydrogen peroxide reaches a certain level, cells stop dividing and begin to elongate. So therefore, the balance between this hydrogen peroxide to superoxide anion is very, very crucial, and it's important for the transition zone. So, so far we can say that it's really important for the ROS into the growth and development. It has also can trigger the plant organ morphogenesis. So the first example that I will talk about here regarding the seed germination. So in the dry condition and dormant seeds, plant embryos and the surrounding endosperm display very limited metabolic activities. So they are dry, so the metabolic activity are really, really low on that case. So ROS production, thus in the speculated there into the dry samples are very, very also very low. It is clearly, we can say that it's very low ROS production over there. But after the seed imbibition and during germination, metabolism rapidly resumes. So, and also the ROS production increased via various pathways. The spatiotemporal correlation of increased ROS production and accumulation during the onset of germination can positively influence the release of dormancy and also the germination. So ROS level increase after the seed imbibition. Yes, because it started, all the metabolic pathways has been started there, and also it acts as a positive signal for germination. However, I should mention here that above after certain limits, ROS are either too low to allow the germination or could be too high and affect the embryo viability and therefore prevent the delay or maybe sometimes delay the germination as well. So I would say the ROS homeostasis here during germination needs to be highly controlled or very tightly controlled. So here you can see there are some genes which are really involved into the pathway, and basically ABA and gibberellic acid pathway, they are responsible for the seed germination, and here ROS plays a very important role. On the other hand, I will talk about the floral morphology as well. Because there are some reports where they have presented there that ROS has really important roles in case of the male gametophyte development, also female gametophyte development, sepal and petal formation, and also about the fertility. So here are the genes, those are responsible for male gametophyte development. Here are the genes for female gametophyte development and also petal and sepal development. So there are some genetic analysis, and it was observed that loss of function of any of these gene or some of these gene could be really detrimental and can also resulted into the abnormalities during the flower formation. So maybe there are problems with the male gametophyte development, also the female gametophyte development. Also there are loss of fertility that happen. Also pollen tube formation was being challenged because of the loss of these genes. So with this we can say that the plant organ morphogenesis is also somewhat dependent on the ROS trigger. The interplay between ROS and epigenetic modification landscape contributing to the stress. It is a very new topic to discuss, and also very few experiments and few papers are coming up regarding that. But this is very interesting. This is very interesting in a way because the epigenetics are the new science now. So it's very important to know about it. So stress-inducing factors modulate the level of ROS, which affect the epigenetic landscape by modulating DNA modification, histone modification, non-coding RNA expression, and also ATP-dependent chromatin remodeling. So this will subsequently affect the gene expression or gene expression patterns in the nucleus and mitochondria, which can contribute to the stress response. Although the underlying mechanism and consequences of reactive oxygen species in this ATP genetic modification are still not completely resolved, and there are much more studies needed to really comment on that. But still, it's a very interesting topic. In case of plant immunity and stomatal closure, there are some reports about the ROS production, reactive on species. They can be really play some role there. So the first example I would discuss about into a plant pathogen interaction between the fungal cell and into the plant cell. So Wang et al. Actually showed a very nice model of inter-kingdom ROS signaling, where in the interaction between a fungal pathogen and their host plant cell. Plant cells generate ROS in the plasma membrane and from several intracellular organelles upon pathogen recognition. So after it is attacked by the pathogen, they activate their this signaling pathways, and also it generates ROS into the system. In the meantime, in the fungal hyphae, produce ROS by then this NOx complex, mainly localized into the plasma membrane or endoplasmic reticulum, which stimulate an oxidative burst response within the pathogen. Scavenging system composed of enzymatic and non-enzymatic system synergistically function to maintain the intra and extracellular redox homeostasis in both plants and pathogen. On the other hand, if I talk about the stomatal movement, I'm not really happy about the picture, but you can see only a few of the genes which are responsible for the stomatal closure, but there are involvement of ROS here. So still I will try to explain it. Stomatal movements are basically controlled by complex signaling networks that respond to environmental and endogenous signals. So regulation of stomatal aperture requires coordinated activity between reactive oxygen species generating enzymes, signaling proteins, downstream executors, such as ion pump, then transporters maybe, and also plasma membrane channels that control the guard cell target pressure. So accumulation of ROS in the apoplast and chloroplast is among the earliest hallmark of the stomatal closure. So when plant really sense and the ROS and it triggers the signal, so plant cell basically close the stomata. So that is also a first signal of the stress that perceived by the plants. So, but even I must say that full understanding of the mechanism underlying between the stomatal closure and stomatal responses and plant pathogen interaction will be required because this is very, very early to really comment on that. But there are significant results or indication that reactive oxygen species play a significant role in plant immunity and stomatal closure. So far, I discussed about the positive sides of the ROS. That means when it really works as a signaling molecule, so it really contributes for them in a positive way. And that time, the accumulation of ROS is not that much high, so it works as a signaling molecule. But I should mention here that the scenario can be completely opposite in the situation where plants perceive or undergoes different abiotic and biotic stresses. So probably, and I hope that we all know what is abiotic and biotic stresses are, but in general, heat, light, drought, any kind of physical stress, I would say, those are related to the abiotic stress. On the other hand, the biotic stress really need an organism and any pathogen or something that can really harm the plant physically, and that is related to the biotic stress. So what happened into the abiotic and biotic stresses? So mostly, as I mentioned, the photosynthetic machinery and accumulation of the ROS into the photosynthetic machinery harm in case of the abiotic stress. Also elevated transpiration and decrease of carbon dioxide fixation, cell membrane integrity, lip senescence, increased metal concentration, hormonal imbalance, and reduced nodulation and nitrogen fixation, and some other options are also there. So in case of biotic stress, the symptoms or maybe the effects are a bit different, but ultimately they will suffer the same kind of effect. So reduced shoot development you will see, then mostly susceptibility to the disease, a damaged photosystem and reduced photosynthesis, then also inhibit the root growth and impaired cell division, then reduced plant biomass, hormonal imbalance, etc. So there is this cartoon or this figure, I would say, is very famous into the stress biology because it is very simple. but yet very descriptive to show the actual scenario into the plant system. So here you can see in the normal condition, the antioxidant and ROS, they are in a equal control. So there is a equilibrium presence between the antioxidant and oxidants into the plant cell. So it is an redox equilibrium is present. But in the oxidative stress condition, or the problems occur when the concentration or the amount of ROS accumulation increase into the plant cell. So this equilibrium that breaks and it causes the oxidative stress. This can happen also in the case of depletion of antioxidants. So it will harm in the same way. That means the equilibrium can be imbalanced by either excess of ROS generation and also depletion of the antioxidants into the system. So it is really, really important to get the balance right. So the equilibrium to the plants should be very balanced so that plants really can see reactive oxygen reactive oxygen species will really generate into the plant system very naturally. So it will generate the way, but the antioxidant system should be very effective over there so that it can really scavenge it. But under the stress condition, it is not the normal scenario. So plants cannot really scavenge always. So then the equilibrium or the balance is not in the right condition. So it deteriorates. Later, I will really describe about the direct and indirect ROS effects. So mostly during the abiotic and biotic stresses, sensors, plants or cells basically undergoes severe effects on lipid peroxidation and DNA damages and others. So here you can see it can also change the epigenetics. In case of the DNA, so many things can affect. For example, deoxyribose oxidation, strand breakage, indels can happen. Then depurination or depyrimidination can happen. Protein DNA crosslinks can happen, etc. These all the phenomena will lead to the chromosomal aberration and also mutations. Ploidy level can also be changed. And also in the protein level, you can see the possible loss or gain of protein function can happen. So it will disturb in the cell cytoskeleton and mutic and mitotic disorders. And also it can modify the enzymatic activity, alter the membrane selectivity, and also change sensing and also the signaling system as well. So although at a first glance, you will see that plants only harm the lipid peroxidation and also the protein oxidation, or maybe at the DNA level, but the direct and indirect ROS-induced effects are so many and so huge. So I would say, like in every steps of the plant cell, they can really sense the level of this effect. So there are some defense machinery itself into the plant cell because plant has an inbuilt antioxidant network into the system itself to scavenge this situation. So this antioxidant network is very important and mostly divided into two types. Those are enzymatic components and non-enzymatic components. So enzymatic components are basically superoxide dismutase catalysis, APX, that is ascorbate glutathione ascorbate peroxidase, glutathione reductase, then GSTs, then glutathione peroxidase, etc. On the other hand, non-enzymatic components are ascorbate, glutathione, then tocopherols, carotenoids, and cysteine, sugars, prolines, etc. So, but in today's webinar, I will basically talk about some of the enzymatic components, those showed already into the plants or into the crop system, that they can really affect or they can defend plants in case of reactive oxygen species environment. So if we see the distribution of the main oxidant resources into the plant cell, they are mostly into the main organelles, chloroplast, cytosol, vacuole, the mitochondrial and peroxisome nucleus. It is very noteworthy to mention here that you can see that as we discussed when the source of the ROS, we discussed about chloroplast, then mitochondrion, the peroxisome, and even at the cytoplasm. So which are the source of reactive oxygen species is also have the defense mechanism inside to minimize that. So plant itself have the machinery to overcome the situation. Just it needed the right balance over there. So if I discuss about the ROS scavenging antioxidant defense mechanism, there are 4 different types of cycles. So water cycle, where actually sodium dismutase is the main component. Then it has the GPX cycle, where glutathione peroxidase and glutathione reductase works as the major, play the key role over there. Then ascorbic glutathione pathway is very famous for as defending the plant system from the ROS, where glutathione ascorbic peroxidase plays the important role, and also the catalysis. It has very important role there. So what really happened when plant undergoes the stress? So first they perceive these signals, or this sense the signal, there are some stress and the unfavorable condition. So the osmotic stress or oxidative stress really happen, and the signal sensing, perception, and translation situation occur. So it activates the downstream transcription factors, and the transcriptional control activates the gene activation downstreams. And ultimately, all the gene cascades, those are working in the defense mechanism, they are activated in the sense, and at the end, they all together produce or make a mechanism so that the plant can tolerate the stress or give some resistance against the stress. So there are so many papers, and it is the transgenic approach to overexpress some genes that can really give some resistance in case of the stress conditions are available here. So there are so many papers. Maybe this slide is not very clear to understand, but here you can see so many genes, for example, SODase, APXS, catalysis, then also DHR, glutathione reductase, GPXS. These are overexpressed in different plant systems to minimize the ROS condition. So I will discuss about some examples. So for example, in 2016, Fenatol showed some data regarding the SOD into the tomato, and from the expression profile, you can see here that the superoxide dismutase are really expressed highly into the different plant tissues. And also under the stress condition, in the salt and drought conditions, they are highly expressive in some cases. And from the genomoid in silico analysis, there are basically 3 types of SODs. That means superoxide dismutase, these are iron type, then manganese type, and copper zinc SODs present in the plant system. This gene, that means they are also overexpressed in rice, and they showed better performance, I would say, if you see here, the germination rate and also the fresh weights, then MGA content, and also the plant height, everything. That means all the parameters that are required for to really analyze whether the plants are performing better or not. They showed better performance in case of in comparison to the wild type. So transgenic lines are really good in overcoming situation, and also in case of not only this salinity stress or drought stress, in cadmium stress also you can see that transgenic lines mostly perform very better when the superoxide dismutase is overexpressed. In this paper, it is Liu et al. In 2019, they showed that there's a hydroxyl peroxide mediated oxidative stress in the different cassava plants. Here you can see the wild type plants are really suffering from the oxidative, ROS production. The transgenic lines, they are performing better. Not only in this case, but also when in the biotic stress condition, the transgenic lines, you can say the wild types are not really performing well in comparison to the transgenic lines. So there are something, and that means the SOD is working behind that, so that they are giving some stress tolerance to the plants. There are APXs, as I mentioned, they are also in the front line of scavenging ROS into the system. They are also very important there. So in 2016, there was a paper in PLOS ONE where they showed the transcriptional profiles of APX into the rice. The expression level was high in different tissues, and also the expression of rice APXs under the stress condition, they really performed better. That means their expression level was really high on that case under the stress condition. So later on, when they are overexpressed into the plant system under salt tolerant condition, here you can see they can survive up to 200 millimolar of sodium chloride. And also their root length and total chlorophyll also perform better in case of the transgenic lines. And similarly, into the stomata aperture, as we discussed before, that the ROS, during the ROS condition, stomatal closure is really important. So overexpressed line under the dark condition, they showed the stomatal closure there. And also here you can see there are some parameters that can really describe the condition of the plants in case of condition. So for example, proline content, then H2O2 content, and also NDA content is important to really check the condition of the stress into the plant. Here you can see in comparison to the wild type, the transgenic lines under the salinity stress perform very good. And the H2O2 content is also very low here, and also the NDA content is low. Similarly, this slide presents the similar function, but in case of the catalyst gene, you can see the tolerance level into the wild type in comparison to the transgenic lines. Also, there was a report that is a mutant of catalyst gene and that performed that here. You can say if you knock out or you have a mutant of catalyst, then that is not really good. In you can see here their growth and development are not really up to the mark in comparison to the wild type. So that means the catalysts are really important for the role, and it plays a very important role against any kind of stresses. So, and lastly, I would discuss about the last reactive oxidant species, this antioxidant enzyme that is known as glutathione peroxidase. So mainly into the plant system, SOD and APXs are mostly important, and they work into the front line, but GPXs are always there, but not really studied very well. So during my PhD, we really worked on the GPX proteins, and we checked the expression analysis of rice GPXs under different developmental stages and abiotic stress conditions. So here you can see the expression level in different tissues are really high in case of rice, and here also you can see there, and the expression of glutathione peroxidase under different stresses also showed very high response there. So we overexpress one glutathione peroxidase gene from pennisetum and enhance the phenotypic morphogenesis, and also resist the accumulation of ROS under stress. You can see the transgenic lines are performing better in comparison to the wild types. On the other hand, as I mentioned, the ROS accumulation and lipid peroxidation are the signals for the plant to show whether how the plants are performing. So here you can also see the proline contents, the NDA contents, and H2O2 contents. Transgenic lines are always better showing than the wild type plants. Also, in my earlier slides, I discussed about the correlation about photosynthetic machinery, because this is the first thing that can adapt or could be under jeopardy by the ROS system. So we measured different photosynthesis efficiency, and we found that the transgenic lines are performing way better. Here from picture as well, you can really see the transgenic lines are really green and they are performing very well. We also went through the yield production, and we found this, there was no pleiotropic effect on the plant growth and also in the yield content. So overexpression of GPX really gave some tolerance to the plants. So with this, I would definitely say that, and also from the literature also, what I understood that maintaining a basal level of ROS in cell is essential for proper cellular function. Mittler showed these things very nicely. That means decreasing ROS level below a particular threshold could result in a suppressed cellular proliferation and also can negatively affect the differentiation and immunity. On the other hand, excessive ROS are detrimental. He discussed about 3 levels, for example, the cytostatic level, then the basal level, and the cytotoxic level, and proposed that maintaining a basal level of ROS, which is above the cytostatic level but below the cytotoxic level, is very important. So proper redox biology is essential for cellular process. He also proposed a bell-shaped curve to represent the same function. So that means if the ROS level are too high or too low, that can impair the plant growth and development. whereas maintaining ROS levels within the right range promotes the plant health. So to summarize, I would say that it is an integrated overview of the redox homeostasis into the plant cell. That means plants undergoes different abiotic and biotic stresses that can really produce the environmental challenges. But on the other hand, plant has the antioxidant system itself by non-enzymatic and enzymatic components. So the balance between these two systems is really, really important, and that we discuss about into the webinar, that this balance can really control the hormone production, degradation, or conjugation, and also the signaling, gene expression, stress sensing, metabolic regulation, cell tissue differentiation, or morphogenesis, and also organogenesis. And other was the physical processes, for example, stomatal aperture or closure, cell wall loosening, membrane potential or modulation, cellular number or size, and also plant-organism interaction. So in a whole, the redox homeostasis into the plant cell that can control, and the control is in plant's hand that can really control its situation over there, but it might need some help by the overexpression of some genes, can be because maybe it could be the detrimental, or the stress level is too high to combat the situation, but plant has the capability. So we need to focus on the redox homeostasis very well so that we can help the plant. But there are some questions yet need to be answered. So for example, first one is multiple ROS sensing mechanisms have been recently documented or suggested. However, although indirect evidence suggests that existence of some mechanism, for example, a sensing of apoplastic ROS, remain unidentified. We need to really focus on that part. Number two, in many cases, ROS accumulation is connected to the calcium signals. However, it is not really very clear to my point of view how these stimuli are connected. In this context, we can say that the most outstanding question relate to the identity of calcium channel and their activation mechanism. Number three, there are multiple lines of evidence support the concept of locality in ROS signaling. Therefore, the special emphasis should be dedicated to the special distribution of potential ROS sensors. So that is another thing. So number four, I would say the previously proposed retrograde signaling pathway based on ROS diffusion to the cytoplasm should be re-evaluated. Because the recent documentation of chloroplast to nucleus protein and hydrogen peroxide transport suggested that the cytoplasmic ROS can arise from multiple sources. So number five, although the signaling of function metabolites and their oxidative derivatives begins to emerge, but there are probably many more such signaling molecules that await identification. And lastly, relationship between compartment-specific perturbations in redox balance and also the ROS formation should be studied. And how they affect each other is not always clear and will need to be addressed to determine exactly how they contribute to different signaling pathways. And this concept is proposed by Suarez, and I will definitely agree with this. This plant stress-related studies that is a combination of classical and innovative approaches by applying different morphological, biochemical, and molecular tools can be very essential or should be started into the study so that we can help in a way so that plant can really overcome this stressful condition. At the end, I would like to acknowledge, or I would like to thank the Plant Molecular Biology group at ICGEB New Delhi, my PhD lab over there, to giving me the opportunity to work over, and also ICGEB for providing me the fellowship. I would like to thank my present lab, which is Plant Breeding Institute, Kiel University. Although my work is completely different now, I'm completely working on a different project here, but still, this is really important to study as well. So this is my group here, and they are very nice group and very lovely people. I'm really happy to work here. Then lastly, I would like to thank the Plant Breeding and Biotechnology Lab back in Bangladesh at the Department of Botany, Dhaka University. And at the end, I will like to thank all the members of the Bioingene to giving me the opportunity to focus or share my experiences so far with you all. Thanks for your attention. Thank you very much.
Thank you. We keep some of question in Q&A box. Can you get it? It is 12.
Yeah.
As your ways, you can answer a few of them.
Okay. Okay, I can see some of the questions. Okay. I can see one question, that is, what is the role of ROS in plant disease management? So, as I discussed, only disease management, I would say not really. There are not so much of studies so far, but this is an emerging topic, I would say. So, as I mentioned during the fungal invasion, if you remember the slide, or if you go through the slide again, then you will find that both of them are really having this scavenging system. So both of the system can really sense their signals from each other upon invasion of the fungal hyphae, or maybe some other biotic factors. So scavenging system itself works there as a signaling molecule to activate the antioxidant mechanism over there. Okay, there is another question then, can manipulation of ROS induce gene silencing? It can be, like, yeah, the manipulation of ROS, it is possible. Like, you can really, by manipulation into the ROS, it can lead to the gene silencing as well, because it will harm in a way so that the plants and other, the DNA level and also the protein level can be really harmed in a harmful condition. So it can be related to the gene silencing. Someone asked about the antioxidant good for human health or not. Okay, that's not really relevant to that. Okay. Okay, I think it's fine.
So we can move on the interview section. I hand it over Shoma for that, and I request Shoma to again introduce our speaker, because we separate this section and separately upload it to the YouTube. So just speaker.
Okay. Hello. We are very happy to have Dr. Islam with us today from Germany. For all the viewers who have joined in later, and also this is a separate section, so I'm talking about her now. Dr.Tahmina Islam worked as an assistant professor at the Department of Botany, University of Dhaka, Bangladesh. Currently, she is doing her postdoctoral research in the Plant Breeding Institute of University of Kiel, Germany. Her main research focus is plant stress biology and genome editing. She has completed her bachelor's and master's from the Department of Botany, University of Dhaka. Her PhD dissertation focused on abiotic stress tolerance in rice, which she completed from the International Center for Genetic Engineering and Biotechnology, New Delhi, India. With her work, she aims to develop stress-resilient crops in context of the South-Asian subcontinent, specifically Bangladesh. Dr. Islam, our young viewers would be very interested to know what motivated you to take up plant science research and how you built your career as a researcher.
Okay, so already if I talk about my career, yeah, it's very early to talk about, but still, yeah, I know, like, the target group is different from ours. So I would say, to be frank, I would not lie. At the beginning, I was not really onto the plant biology. I was not really interested, to be frank. I wanted to be a doctor or something, but unfortunately I couldn't pass the exam. But somehow I get into the biology department. And there, one of my professor, when I was in my bachelor's in the third year, he really taught us very well the molecular biology. And that triggers me actually, you know, like that not only we are not only constructed into only the plant system, and also there are so many things to do. So that clicked on my mind, and I would like to mention my professor name as well. He was a professor, Dr. Rakavi Sharkar. He really encouraged us on that moment. Then later on, I did my master thesis with him, and that was the window, I would say. So when I entered into the lab, I came to another prominent scientist, and he is Professor Dr. Imdadul Haq. He was also very encouraging person, and he really motivated me to do the research. So you can say like completely a person who was not really interested to that, but I became much more involved in into the plant system, and also basically now I liked it very much. This is the only system I would like to talk. And later on, I went for PhD immediately after my master's. Before my master's, I got the position for this PhD, and I went there. And ICGEB, it was a very, very nice institute, I would say. On that time, it was really good, and plant molecular biology group was really nice over there. So I found my supervisor, who was Dr. M.K. Reddy, who was very cooperative on the time, and he actually gave me all the opportunity to work by myself. He let me to do the thing that other way I want. So that's how really I get onto myself, not to depend on the others, but to do the way you are.
Yes.
Then, yeah, I would say, so my master's lab and also my PhD lab, and also very nice colleagues over there. They are really, really nice colleagues over there. They really focus. They are really focused on that time. We discuss about science. We discuss about how to do the research. Actually, PhD period is the training period, I would say. You learn so many things. That time you need not to be very, you should be very passionate, but not really, you know, like strict to something that you have to achieve this, because here on that time you are there just for learning. So you are learning and you are trained up for the future. So that training period, I would say, actually completely changed my concept about science, and from there now where I am. So that's it.
Okay, all the best for your future endeavors. My next question is that what, according to you, is the future of genome editing in plant sciences?
Oh yeah, that's a very like important topic nowadays. I can say this is the hot topic nowadays. So also here in Germany, I'm working on the genome editing. So my whole project is based on that. But I would say like every day, if you go through the journal or the publications, you will find there are lots of paper coming every day on the genome editing. So each of the day you will find thousands of people are working there. So that means something really interesting over there. And also it's a big thing. So I would say so far people are really editing in a single gene, or maybe into the low scale, but there are some points that I would like to address here. For example, multiplexing or multiplexing or stacking of several genes. You know, so plant system in several pathways, there are so many genes work together. So maybe change into a single gene cannot give you the expected phenotype that you want. So it is necessary sometimes to really stack or multiplexing of several genes altogether so that you can find. So nowadays, this concept is coming up. So this is very important. And also I would say that I am also working to develop a system so that we can stack multiple genes over there and we can target them at the same way, same time. So this is very important, and this is, I think, up to date. And also, I would say this high throughput mutant libraries is also very important, because there are so many genome sequences available, but all of them are not really analyzed so far. And also it's not possible to go through one by one. It will take a longer time to analyze them, because loss of function analysis is very important to analyze the function of a single gene. So maintenance of a high throughput mutant libraries in a single way would be very efficient in a sense. and also the gene regulation, not only the gene knock in or knock out, and also regulation in a way so that you can change the expression of a particular gene by changing base pairs. Now, very recently, you must heard about this prime editing already came. So that is very much into this time we can discuss with and we can work with in future with the genome editing.
All right. What are your future plans for improving agriculture in Bangladesh?
Okay. Yeah, as a Bangladeshi, I was discussing about with a Shupos, and regarding that, I would definitely go back to Bangladesh. It's my plan. I never wanted to stay outside. So if I will go back very soon, and if I work there, so for the agriculture, maybe I will work with the stress biology, because from the Asian subcontinent, you know that we suffer from various stresses. Although this kind of stresses you will not find in Europe and other places, but in Bangladesh and in the Asian subcontinent, you will find very high quantity of drought and also flooding. We are always under the flood. So this is a very critical condition for us. So I would definitely like to work with those aspects. And also, as I learned how to do the genome editing, also I would like to work on that as well, so that I can really improve some of the varieties or some of the plants, so that we can improve the yield contents as well. So this should be my focus, like with stress biology and also with a combination of genome editing.
Great. My last question to you is that what would your words of advice be to the youngsters who want to take up a career in plant science research?
Okay. Yeah, like I don't consider myself very efficient to comment on that, but yes, like I can tell from my experience that if someone told me when I was in PhD or when I was at the beginning of my career, so that I can share actually. I would like to hear on that time, like if somebody told me those. So I think first thing should be passion, to be frank. Like you should be very dedicated and passionate about your work. So this is very much important. So that is the one thing. Then consistency. You should, because you know every day is not the same. Every day is different. Every day is different from the other difference. So new challenge comes, and you cannot expect that it's a smooth life and also a very softly handed life. It's not like that. You will find a roller coaster in your PhD life and throughout the cell. So you need to be very consistent and need to be prepared for the bumpy rides. So that is to say, you should not lose your hope at the end. And also these two things that I will go to, I will go say that because, and this thing I learned now actually from my professor here in Germany, Professor Christian Jung. He always tell us to ask questions. So it is very important to ask questions, because what I find, normally there are so many questions in our mind, but we don't ask. But it is necessary to ask. And also you should have a very clear definition in your mind when you are doing some experiments, to distinguish between your aims and objectives and what is your expectation, basically. So before performing any experiments and before performing any of the research, you should clearly define what is your aim and what is your expectation. So it should be very clear. And finally, you should love your work. Whatever you are doing, you should enjoy it. That's the main thing. You should enjoy until at the end of the day, you will love it.
That's a great words of advice. We're very happy to have you here today. Let's get back to Shubho now.
Thank you, Dr. Islam, for your elaborative talk. I am sure so many appreciation I have in YouTube that your talk is very nice, and so many people are able to connect it because it's presented in such a way and in such an informative and easy presentation. So I hope in future we get you with your new research, like now we working on CRISPR. So we hopefully in future we again we met in this platform to discuss your CRISPR journey. And definitely it will very helpful to Bangladesh and Asia if you come and do your further work, because you are early in the career and you have so many times and you learn so many modern sophisticated techniques from there. So it will help Asia and all over the world, because we are common to one thing, that we all love plants and we all love plant biology. So I very thankful to you that you met this day, and we from the behalf of Bioingene, it's our pleasure to showcase you and your research, and your interview also. I am sure motivated many people, including Bangladeshis also. So you leave this meeting today. We have some unfinished due to some technicality, how to apply for the certificate and all. And in our Bioingene website, I think by tonight you got the whole list. Those are the giving the feedback on your views on your webinar. So there is a very interesting to you, I think, because you got the actual listener and their countries, their institute, their names, and they all got the certificate for participating in. So hopefully, best of luck for your future, and again, we shall meet one day. Thank you.
From my side, thank you very much. It's a pleasure to give a talk over there. And yeah, I definitely would like to, if you ask me again, I would definitely like to give any lecture there. Thank you all of you to have this platform for us to showcasing our work or our view. Thank you very much.
Thank you. And
I will leave. Bye.
Bye. Thank you, Neha, for joining us. I know your health condition, but okay, thanks a lot.
Yeah. Okay.
Shoma, please give some advice to the certificates and others procedure, our upcoming webinars. And tomorrow we have another webinar, so go for it.
Hello. Yes. Okay. We have a webinar tomorrow by Dr. Deepak Gayan. The timing is 11 a.m. For today only it was 12 a.m. For tomorrow it is again at 11 a.m. So please remember the timing. And we're looking forward to his talk. I have some information. It's all information actually. You all know. Some of you don't know, so I'm telling you again that you can apply for a certificate. You can do that by clicking on the feedback link, or you can also visit our website. You need a password that is already provided after the presentation. Get that password and fill up the feedback form. And that's how you apply for a certificate. And regarding actually downloading the certificate, you have to visit our website. Visit our website and scroll down to the bottom of the home page. There you will find all the previous webinars have been enlisted there with their title, the speaker, and a certificate link at the end. So there are actually 2 links for each webinar. The first link, that is the title of the topic of the webinar. If you click on that link, you will see an Excel sheet open. There is a list of all the applicants for the certificate. So if you have applied for the certificate correctly, you will find your name there. It is a long page because many of them have applied, almost like 1,000 to 1,500 people on an average apply. So it's arranged alphabetically. So scroll down or find your name, check that it is your name and university. And on that line, right next to your name, you will find there is a file number. You will require that number because to click when you click on the certificate link, then you will find that there is a you will straightly go into the Google Drive folder. All the certificates have been issued there, and the PDF files have been named with the file number. So your name has a file number right next to it. You find that file number, that is your certificate. Download that PDF, that will be your certificate. Another thing I want to say is that because it is a large folder with many PDFs, nearly more than 1,000 PDFs, so definitely it is going to take some time to open, for all the PDFs to show on your screen. So if you're impatient, you may not find your file number because the page has not loaded yet. Give it some time. And also it would be better if you have a good internet speed, because otherwise it'll take a lot of time. But still, have patience, wait for some time, find out your file number, because if your name is on that list, that Excel sheet, then definitely your file number is in the Google Drive. There is no way to miss that. So download your PDF and there you go. You got your certificate. It's so simple. Another thing I want to say is that for uh, this whole process, we will need some time after the webinar. Like the webinar is ending now. So we will work on this. We will see all the response sheets. We will see and we will make a list and then we'll make the certificates. So this process takes some time. So don't expect that right now you're going to get your certificate. So it's not possible like that. So it will take some time, maybe by tonight or tomorrow, you will find that the link is there with the topic of the webinar and the speaker, and then there's a small link for the certificate. So you have to wait until later today or tomorrow. So within 2 days, you're receiving your certificates. I think that's pretty much it. Let's go back to Shubho now.
Thank you everyone for joining us, listening the webinar. Tomorrow we have another webinar. So I'm requesting all of you, our online registration is ongoing from our website. I already mentioned our website here. Otherwise, it's just type bioingene.com. It redirects to our currently working website. Go there and fill your registration form because it's mandatory. And feedback form also the mandatory. Those are come in the later in this webinar. I'm going to give this link and password for the last time, and we will end that meeting. If you are able to fill the feedback form, your name should be in the list. If your name in the list for the applicant, as the applicant, your certificate within the shared Google Drive. So don't mail us to search your certificate, because it is your duty to search it and download it. We make it and upload it for you for download purposes. So we are not going to search your certificate on behalf of you, and we are not giving you a certificate by individual mail, because we did this in first and second webinar, and so many requests we also getting till date that they don't get the certificate. So we can't do this mistake next time. So this is our protocol. You can register yourself to any of upcoming webinar or all of the upcoming webinar. There is a lot of webinar in our Bioingene platform. Many more will be added very soon. Register it. The listening or watching webinar is easy. Subscribe our YouTube channel We broadcast our webinar here. After the watching the webinar, you have to fill the feedback form. It is mandatory to apply a certificate. When you apply the feedback form, you need a certificate, you need a password, which will given that time during the question answer round or within any point of view during the live webinar. Feedback form is activated for maximum 2 hours. When it's when it gives to you, and between the closing and starting, it's about 2 hours the activated in our website. So within that time, you have to submit your feedback. Tomorrow we have a webinar on improvement of storage stability and productivity of rice through genetic engineering. It will be delivered by Dr. Deepak Gayan, Central University of Rajasthan, India. So I'm just for the last time giving the link and password, and we'll end the meeting. I'm requesting all of you keep in touch in our Facebook page, in our YouTube channel, so we can connect very easily to you. Bye-bye.